or solve question related to the osmosis first of all, because these are quite important, and moreover there are questions related to these one in P4, like in which you have to design an experiment. Okay, we'll discuss that paper as well because you have to give that paper as well. Okay. um no, there are certain practicals, so this chapter is important with respect to that. Have you performed the potato stick practical? Yes. Okay. What? What have changed or not? Okay, 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 okay. Okay, got it. So no, I'm not talking about that. Just give me a minute. Why it's not selected? Okay. Um. Okay. No, I don't know what's the issue with this one. चल रही है? Okay. So I have a question related to a potato stick basically. So you would be you must have given certain sticks and you were asked to put them in certain solutions. So here we have three solutions. Okay, point 0.1 mole per cubic decimeter. Then we have 0.5 mole per cubic decimeter and 1 mole per cubic decimeter. Let's consider this one as hypotonic. Let's say this one 1 mole per cubic decimeter as hypertonic, and this one as isotonic, though this is not isotonic, but let's consider this one. Okay, then we place the potato sticks for 5 minutes into the solution, and then we'll observe the changes. Now the changes are related to that, though this is like a whole question. You have to design the experiment and then you have to tell the results. Okay, so how would like if the question is how would you design an experiment to check the osmosis in plants? Experiment. Yes. Let's say 6 marks. Yes. Okay. Equal concentration of salt inside and outside the cell. Okay. Okay. So the very first thing is now you have to note points because you have to write certain things. Your number one point should be like what is the dependent variable and what is independent variable. Okay. Now how do we define dependent variable or independent variable? That one is independent variable. Okay, that is controlled by us. Okay, so here the independent variable is the concentration of solution, right? Because we are changing the concentration of solution, right? So this one is independent variable. So what would be dependent variable? The size of potato sticks. Okay, dependent variable is dependent upon the independent one. Like if we are changing the temperature, what would be effect of on diffusion or what would be effect on certain thing, that would be dependent. Okay, independent is the one that we are changing. So we are changing the concentration, So that would be dependent variable, independent variable one, and with that, the size of potato stick would change. That would be dependent variable. Okay, so how to write this? So when you write this, you would be like writing it like this. Okay, yes, no, not necessarily. Maybe if you increase the temperature, the other thing would decrease. If you are checking the effect of temperature on enzyme, the temperature would be independent, and the rate of activity or rate of enzyme activity is dependent one. Simple. Let's apply it to the simple one, rate of photosynthesis. If you increase temperature, the rate of photosynthesis would increase, right? Yes, yes. And the rate of photosynthesis would be dependent. Clear? So how to write this? Definitely we are not going to write like this. We would say that the solution of two different concentrations would be taken. Okay? This is your point number one. Mean mark would be given on this point number one. And point number two be what would change. And the size of potato stick, or sorry, or the change in the size of potato stick would be observed. Yes, that one is dependent. So we are mentioning independent and dependent. Okay. The solution of two different concentrations, I haven't mentioned different. Two different concentrations would be taken and the change in size of potatoes would be observed. Okay. Now the third important thing that we need to mention is the constant things. What are constants? What should be okay. The no, the concentration is the independent one. We are changing the concentration. The size of potato stick should be same. The initial size of potato stick should be same. No, we have to mention all, whatever is required, as many as we can. Okay. Second thing, time for which these are immersed. Yes, these sticks are immersed in solution. Okay. And moreover, if there are plants, then definitely there is more. Okay. Third thing, the temperature of solution, it should be constant, because if there is higher temperature, then there would be like it would make a difference. There was a question related to it, why you cannot observe osmosis in boiled carrot sticks. Why? Carrot sticks. Hmm. But if we boil the temperature now if yes, the exactly the not as well as enzyme denature, the structure of cell wall and cell membrane got disturbed, so there won't be control of movement at all. You are basically denaturing the cell membrane as well and cell wall as well. The cell membrane is the one which is controlling the movement. Okay. Okay. Denatured. Denatured. Done. Okay. Now these are the 3 points that would be given on the basis of what how you are designing the experiment. Okay. Now let's carry out the experiment. How to carry out? We first, the initial mass of potato stick was calculated. Yes, yes, you need to know the experiment. Using what you have used, weighing balance. This is for P4. When you have to design the experiment, you have to learn like what things are required. They are not much. Yes, it will be like this. There are almost 10, 12. You can learn those. If mass of stick was calculated using weighing balance, the change in mass after immersion, 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 what is the word they have used here? So most of the time the word is given in the in the like question statement. You can choose from there. After immersion would be recorded after recorded after a specific time, whatever time. If you have selected 10 minutes, or if there's no need to mention the time, 10 minutes or 5 minutes, after a specific time or after a given time. Okay. Now this is how you carried out experiment. This is point number 4. Now point number 5 is this would be repeated. This step would be repeated for another concentration. We have mentioned in the start that we have taken solution of different concentration. The other concentration, and the readings would be recorded. Moreover, one more thing, you don't have to write I or U or V. Just write it in the passive form. A reading would be recorded. Okay? 5 points are completed. Now, in the last, you might have to give the safety precautions. Like what precaution is required in this one? Number one, in this experiment, because you are cutting something, gloves are mostly used when you are using certain type of chemical. For cutting, for cutting, always cut the potato sticks or any, even if the plant is given, this is also part of plant. Cut it away from the body. ठीक है, away from the body. Second important thing, cut cut the potato stick away from the body. Simple. Okay. If temperature is required, use water bath. If you are checking the temperature one, I'm telling you these general ones. If temperature is more, use water bath. Do not directly heat. No, if temperature is required, you are checking the effect of temperature, like of 100 degrees Celsius or 60 degrees Celsius. Definitely the temperature is really high. You cannot touch it directly. So you have to use water bath, or do not heat directly. Okay. If you're using any electrical appliance, for example the electrical lamp in photosynthesis, okay, keep it away from water so that you won't experience any shock. Electrical lamp, this one is part of photosynthesis. Okay. So these are different precautions that you need to keep in mind. Or if you are using chemical, use gloves. If a certain gas is produced, then use goggles. Goggles in case of gases or vapors. This one is part of chemistry most of the time, the vapors and gases. So we use goggles at that time, or gloves for chemicals. Okay. These are the certain precautions that we follow. So here we can write it for cutting, like cut the potato sticks away from the body. One more thing. There was one more for starch test, for food test. There is a precaution, food test. Always use, like for starch one in which you have to put it on a white white tile. Exactly. Why do we use white tile? So that we can easily observe the color. Okay. Have you got it? How to attempt a 6 mark question in the ATP? Yes. Okay. Mention um, the independent variable, dependent variable, the constant, the instrument that you have to use. Then the repetition is very important. Okay. And the safety precautions. Okay. So this is the how you would design an experiment. Now the question related to these questions, if there are certain questions, predict which of the liquid. Let me again just copy the solutions. Okay, I'm giving you solutions here, here, there, here. In which of the solution you would observe the largest decrease? So this the mass becomes smaller or the size becomes smaller in hypertonic. So which one is hypertonic? In which hypertonic mean more concentration. Yes, 1.0 mole per cubic decimeter sodium chloride solution. Write the complete statement. Okay. Okay. The student dried the potato stick with paper towel before putting them on the electric balance. Why? It's correct. To remove the excess water. To remove excess water. So that it won't disturb the readings of your experiment. Okay, explain why the potato stick had become soft and floppy. Wait, we have written over here there is a largest decrease in the mass. There is decrease in the mass, lowest mass. So what would happen if the mass lowered? Yes, you have to tell the complete process. There is okay. There is let me write. There is less water outside. Okay. So water due to osmosis, water would move out of the cell. Okay. 2 marks. What would be your third mark? Now related with soft and floppy. Good. The turgor pressure is lost and cell would be plasmolized. Turgor pressure is lost and cell shrink. Sorry. Sorry. Due to plasmolysis. Okay. Done. Now again the question is here. The student followed the same experiment procedure with boiled potato sticks and found no overall change. Why? So no net movement. So there would be no net movement by osmosis. Okay. Okay. Now there is one more practical in this chapter because this chapter is more important for ATP and this paper 2, both paper 2 and 4. Okay. This practical is related to whisking tubing. Okay. It's a whisking bag. What is this? This is basically whisking. What is this whisking bag? It is a semi-permeable thing. Semi-permeable membrane. Yes, yes. So we just placed a test tube, rather, no, we have placed this whisking tubing into a test tube. Okay, and this test tube contains water. Clear? So this means this one is a hypotonic solution, right? Exactly. Is this thing clear? Now, this whisking tubing has starch molecule, mean there is lesser water inside. Is it so? Lesser water potential inside the whisking bag. Okay. Now we have to tell for 4 marks. After 30 minutes at the room temperature, the content of the whisking bag was stained blue black because we have used iodine, but the water outside remained yellow. Okay, let's just figure out what they have told. Yes, the student added some iodine solution. Now here we have iodine as well. Iodine is actually yellow brown in color, right? Iodine is yellow brown in color. Have you seen pyodine in your home, right? You have used pyodine. It's the tincture of iodine. It's brown yellow in color. Where is starch? Where is starch? Inside this one. But starch is not moving here. Why? Wait, wait, wait. There is iodine solution in the test tube, but the whisking bag contains the starch, right? Now the whisking bag got colored, but this test tube doesn't. Exactly. Why? Why it's not present? Because okay, why it's not moving out? Starch is already less in water. There is no starch in water, so it should move out due to concentration gradient. But starch is not moving out because it is bigger in size. This is a function of okay. Yes. Hmm. Okay. Okay. Let's see. Whisking tube is like a balloon. We filled it with starch solution. Okay. Then we place this balloon into a water of a test tube. So if it is partially permeable, the starch is not going out. Why? Due to its size. So this we have to mention. Okay. So the experiment is explain your result. Explain mean you have to give reason why they are not coming out, why they are getting stained inside the whisking bag but not outside. So we would say that iodine molecules, these are smaller in size, diffuse into the whisking bag or whisking tubing, um, smaller in size, diffuse into whisking tubing, okay, where starch is present. We have added iodine in water. It diffuse into the whisking tubing due to its smaller size. Okay, so it would be stained or it would be stained blue black or turn blue black. Okay, however, now on the other hand, starch molecules being bigger in size, starch molecules are too large to pass through whisking tubing, so they are not diffused out. So wouldn't diffuse out. Okay, if they won't diffuse out, so your water would remain yellow. Okay, now when we are saying that iodine can move into and starch can move out, we can also tell here there is another point that the pore size pore size of whisking tubing is suitable for the movement of iodine molecule only. I mean, it is so small that only iodine can pass, starch cannot. Starch is a bigger molecule. Okay. Shall we move to the next question? Movement of molecule through the membrane. Okay. Um, have we discussed this yesterday? Like, what are the factors which increases or decreases the rate of diffusion? No? Yes? Okay. So, what is factor number one? Steep gradient. Steeper the gradient, more will be diffusion. Is it inversely proportional or directly proportional? Direct. If we increase the temperature, the rate of diffusion also increases. Why? And they move faster, diffuse faster. Okay. What is third one? That is part of steep gradient. Surface area. There is another one. If there are more marks, there is another one, which is called distance. If the distance is lesser between the like two gradients, then there would be faster diffusion. If the distance is more, then there would be slower. ठीक है? Okay, slower diffusion. Shall we apply? I guess you are going to learn. Okay, now how would you design? Now you have learned the whisking tubing experiment. How would you design an experiment if this is part of your P6? If it's a P6 question, design an experiment to show osmosis through whisking tubing. Wait, wait, wait. No, here we don't have to use the concentrations because we are given with whisking tubing. Let's design an experiment related to whisking tubing and osmosis. Okay, this is our experiment. Let's figure it out. Okay, we are not going to use these terms. We have to write it in the form of sentence. That is already whisking tubing. That is whisking tubing, na? Osmosis would be dependent on whisking tubing. Okay, so we'll say whisking tubing of a certain pore size. Yes, you have to. Pore size would be taken to check this one. Okay, to check the rate of osmosis or to check osmosis. Okay, okay. Second thing, now you have to tell how you're going to do this experiment. This is like the whole, like this question that we did. Now, what would we do? We add start solution in the whisking tubing. I'm using VT, okay? You don't have to write VT, you have to write the complete words. Okay, I'm using VT for like short form. Okay, we add start solution in the VT and place it in a test tube containing, let's say, distilled water. Okay, why I'm writing distilled water? Because in while doing experiment we usually use distilled water so that it won't affect the readings, because the other water, the tap water or the mineral water, contain other minerals. Okay, distilled water. Now just check. I have mentioned the dependent variable, independent variable, and how which instrument I'm going to use. Okay, so this is how you design your experiment. There's no need to tell them, okay, this is the apparatus that I'm going to use. Just go, like, go smoothly. Hmm. No, no, no. Whisking tubing of a certain pore size would be taken to check osmosis. Exactly. Okay. Then we have mentioned, um, we have used test tube, we have used this whisking tubing, we have used star solution. Okay, we are telling them everything. Okay. Now, uh, place, or sorry, huh, or pour. Let's no place. Place few drops. What do we say? I forgot. Not pour. There's a word. Place few drops of iodine solution in the test tube containing distilled water. Okay. After a certain time, or after a given time, observe the changes. Observe the changes in the color of test tube and whisking tubing. Okay. Now you don't have to give them result. They are not asking for result. They never ask for result in the experiment design. They would ask for for the method that you have used. Okay. Now what is the precaution that we have to use over here? Because we are using. Okay. Yes. Use gloves. Okay. No, no, don't mention this word. No. Hmm. For what? It's correct. It's correct. For solutions, huh? Exactly, for solutions. Okay. Now there is one more precaution. Listen, while you're using whisking tubing, have you seen the whisking tubing? No? Okay. Um, how to explain it now? Yes, it's related to, like, it's just like a very how, it's a type of test tube, but of rubber, let's say. Okay. Now, listen, we have to bind it with elastic band, and most of the time we use thread, so that this elastic band wouldn't open. Otherwise, it would open. Okay. So we have to tie this whisking tubing really well. Whisking tie tightly, tightly tied. Why should be tightly tied? So that star solution won't come out from the, like, from the mouth of it. Okay. Otherwise, if you squeeze it, the star solution is going to move out. Okay. It's tightly tied so that star solution remain inside. Remain inside. Okay. Use a white background to observe, because here colors are involved. Change in color. Okay. You shouldn't be using black wall. Clear? I'll send you this document, don't worry. Okay, these experiments are also given in your book. You can learn from there as well. Okay. Okay. Let's move to the other question. Okay. Now there is another question which is related to the active transport. Okay, now listen to the you have to listen to the question really well because we already did this type of question yesterday. Some plants can be grown in water using a technique called as hydroponics. These the roots are immersed in water containing a solution, and some ions can be absorbed both by diffusion and active transport. State two features of diffusion that do not apply to active transport. Okay, listen to the question. Read the question carefully. You have to tell about diffusion only. There's no need to write while in active transport this happened. There's no need to write that. Just write it for is good. The the movement of particle. Okay, down the concentration gradient. Okay. What is point number two? What are the difference? That we don't need to mention. We don't have to tell about active transport. We have to tell only about diffusion. What is present in diffusion but not in active transport? What else is present in diffusion? Yes. Now the second point. What should be point number two? Exactly. Good. Does not require energy. Okay. So we did such question yesterday about the photosynthesizing plant and bacteria, right? So you have to tell about the photosynthesizing plant only, not about bacteria. I'll send. I'll send. I erased yesterday's one, but I'll share the video link with you. But I'll give you this one. Okay. Then there is the next question. How are roots adapted to absorb ions? Roots, roots. What is present? Exactly. Root hair cells. Okay. Root hair cells. Good. 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 It's correct. It's correct. Exactly. Exactly. Okay. And what if it has to take up ions with the help of active transport? Then mitochondria. Exactly. Through. Again, you're missing the same thing. Through. Exactly. Aerobic respiration. Exactly. Okay. Okay. Uh, what? Let's move to the next one. Okay. Now a graph is given. This important to complete the graph. Okay. This we can do only in like when we have a hard copy. We cannot do it. Perform it here because we have to mention. Okay. I'll add it over here.